Okay, so I'm Vincent Prié. I'm a researcher associated to the National History Museum in Paris and also to CBO in Portugal, and currently working in SpyGen, which is a private company specialized in environmental DNA analysis. And I will share with you recent advances in freshwater molluscs, freshwater mussels, especially distribution, conservation, and taxonomy, seen from an eDNA point of view. So as you probably know, there are many freshwater mussel species, 45 species in France. They are hard to find, difficult to identify, but they are ecological engineers and they bear conservation issues. So in good old times, freshwater mussels would make up to 90% of the bento mass. They filter, the large species are supposed to filter like 40 liters per day and per specimen. So you can see on this picture that it will represent quite a lot of water filtered by the freshwater mollusks. And this filtration results in clearer waters, which helps the macrophytes to grow, which helps the macrobenthos, the insect populations to be more important. And it even has an importance for fish. Like it has been shown like that rivers with a lot of freshwater mussels have many more fish abundance than rivers that lack freshwater mussels. And also mentioning that shells are used as shelters for insects and small species of the macrobenthos. So they have a really important role to play in ecosystems. Some species are highly endangered. This is one of them, Pseudonio auricularius, a large species that used to live also in England. There are some fossil records in the Thames River. It was spread in all large rivers between Denmark and Italy westward, down to Portugal. And it's only known now from 6 populations, 5 in France and 1 in Spain. You have those red dots here. And the species still reproduce, but doesn't recruit, meaning that we don't have juveniles, or very, very few juveniles. It's only very old specimen. They are thought to live for about 80 to 100 years old. So we only have those old mammoths on those populations, and in probably a century or so, there would be almost none left. So this is for one of them. This is an example from Portugal, where they had a big survey in 2002, 2004, and did it again in 2022. So this is quite recent. And you can see that most of the populations were extirpated in this about 20 years last time. They are hard to find, especially in large river, in the plain, in the downstream ecosystem where most of the biodiversity is to be found. So you can imagine this is the Loire River. It's turbid, it's deep, it's navigated, and if you want to find a small species like ordinary Pisidium matacerianum, it's particularly challenging. Even for large species, this is the kind of condition we are used to dive in. So you can see that the visibility is very low. There are plants, and what you want to find is these small apertures of a mussel buried in the sediment, which size is probably less than a centimeter. So they are hard to find. This is one of the challenges for freshwater mussels. They are also difficult to identify, which has led to a big taxonomic confusion, especially in the 19th century, where every form has been described as a species. And for example, Locard in 1893 considered that there were about 500 species in France. A lot of them has been synonymized by Germain and by Falkner more recently, but all these authors still kept a lot of subspecies, which show the difficulty to identify and to separate species. This was the beginning of my PhD. So I managed to do molecular analysis of more of the species known based on topotypes mostly. And it led to a taxonomic clarification. So we now know how many species we have, although some of the subspecies can be discussed. But still, as you can see on this picture, here you have only one species and many morphs. So we still need barcoding to identify properly some of the mussel species. So we still need to collect a live specimen. It's invasive. You have to take a piece of tissue, and it's impossible to implement on large scale. Well, 10 years ago, there was this environmental DNA revolution. So it was first aimed at amphibians and then fish. And when I saw that, I thought it was the solution of my two main problems. It secures the identifications because it's based on barcodes, and it has an increased probability of detection. So it was really a novelty for me, and we managed to develop primers specific for freshwater bivalves, and it worked very well. This is an example of the result we had at the beginning. So in blue, you have surveys done with traditional methods, and big surveys, like many malacologists, divers, many days on each site. And in red, you had what we had with eDNA when we came back on those sites and did eDNA to try to find out if we can find the same species that were observed by the malacologist. And you can see that in almost every case, eDNA was better, especially here on the right, where spherids were considered. On the other samples, it was only unionid, so large species. And when it comes to spherids, it makes a real difference. In some cases, eDNA was not as good as traditional methods, for example, here in Mézières. This is due to the fact that two species were found as shells only. And of course, environmental DNA doesn't detect shells. It detects only live specimen. So these two species are extirpated, but we still have shell data. I will come back to this issue a bit later. So what is environmental DNA? Each of us are going to lose today 5 million skin cells, and all of these, which makes 80 kilo in a lifespan, and all these cells have our DNA. So on the paper, you should be able, from water sample, for example, or soil sample or air sample, to find the DNA of all the species that live here and around the sampling site. This is the theory. How does it work? So you sample some water here, for example, on a river. You extract the DNA from a filter. There's an amplification, sequencing, bioinformatic analysis, because the machine will always give you like 25 million sequences. And out of there's a lot of rubbish, or there are things that you don't want to have, like bacteria or other DNA. So you have a big filtration process. And then the results, which are in this form. So there are amphibians on the pictures, but the results I show are for mammals, for example. And so you have for every site detection or not of the DNA of the species. Of course, this environmental DNA is rare, it's diluted, and it's degraded. So you have very short fragments. The DNA is cut into short fragments. So you have to find primers that will be able to identify every species on the very short fragments. You don't have the 600 or so base pair like for the CO1 that everybody uses in taxonomy. Here, for bivalves, for example, we have 150 base pair fragments, which is quite short. And you have to target places in the genome where you can find good resolution for every species with a short fragment. And you need also high detection standards. So I would last a little bit on that. So dedicated primers, we managed to make them. I mean, it's a computer work. And the primer we have for bivalves are able to identify every Western Palearctic species. So we are very happy with that. And then the field. So on the field, you have to optimize the sampling protocols. So we advise to do twice 30 liters per site, which allows an anti-relative sampling. I will illustrate that later. and optimized lab protocols. So we do 12 PCR replicates, and the coverage is twice 300,000 sequences, which, why twice? Because we have two sets of primer, one for the Unionida and one for the Venerida, which includes the spherids and Corbicula and Dracena. So this has a cost, of course. If you multiply the number of PCR replicates or the coverage, it costs more than if you do smaller things. But with that, okay. Maybe I will come back to that. So if you imagine different requirement, like R1 would be you want to record the diatoms or zooplankton or bacteria. For that, you don't need large number of large volumes of water because you want to characterize the abundant species. So you want to compare that like bacteria composition from, I don't know, a river to another. R2 would be, for example, fish monitoring, stock estimations. Same thing, you will target some DNA that is less abundant in the environment, so you have to have a big higher standard protocols. But you want to characterize the abundant species, you don't care about the rare ones. And R3 would be the rare ones when you want to detect species that are hard to find with other methods. So rare species, early detection of invasive species or viruses. And for this, you need optimized protocols. And for freshwater mussels, of course, we don't want to know that Corbicula is in a river because we already know that. We want to detect if rare species are still present or not. So we push all the sliders up to be able to identify the species. In the field, it means, for example, that you have to make transects from bank to bank to be able to integrate the different ecological conditions. And for this, you need large volume of water. You need high capacity filters because you are going to filter for 30 minutes twice. And you can't, of course, do that with a very small filter. So you need those big filters, which cost a lot also. So everything has a cost. But if you want to be efficient, I think you have to optimize all those protocols. This is to illustrate the reason why we need to do bank-to-bank transfers. I love this study. They had cage fish here in green, and they did eDNA samples for up to 5,000 meters. And you can see that eDNA follows the flow, which was expected, but it's nice to show it, which means that, for example, in this case, if you sample only on the middle, you will not detect the cage fish because they are only found by the eDNA sample that have been done on the banks. So you have to be integrative. You have to really go from bank to bank if you want to characterize the species present in an ecosystem. And another thing that is interesting there is that, for example, on this bank, on the red one, they didn't detect the fish caged 5 kilometers away. Only on the other bank, on the green side, they detected it, but with very, very low amounts. So this gives us an estimation of the distance of detection in a large river. And so it's about 5 kilometers, not more. And this is very important because it's a grain that has a meaning for ecological studies. You don't want to know what's present on 100 or 200 kilometers of river. You want to have a grain size that is quite local. And from our experience, it's probably even less than 5 kilometers in most cases. We do 2 replicates, and here you have an illustration of why. So this is a model of what we would get in probability of detection with 1 replicate. So, for example, in this species, Pseudoneo auricularius, you see that it's between 0.5 to 0.7% detection probability with 1 replicate, and it's much better with 2. So the 2 replicates are illustrated with the red dot. And with 2 replicates, we are around 80 to 95% probability of detection for most species. 2 replicates means 60 liters of water, twice 12 PCR replicates, and etc. So for freshwater mussels, to be efficient, you have to use very optimized protocols. This is the take-home message. high capacity filters, a lot of water, as much PCR replicate as you can afford, and coverage that has to be consequent with the species you want to study. So, for example, with marine fish, the coverage is up to 1 million sequences because the DNA is very diluted. Okay, now some results. So this is what we have in France. So as you can see, we have many sampling sites for freshwater mussels now, with some orphan regions that we are trying to complete. We have about, I think it should be about 1,500 sites now in France with quite a nice coverage. That allows to map the species distributions, considering that we have a good detection probability, at least much better than with the traditional methods. And eDNA provides an up-to-date distribution of live population. This is important because when talking about mollusks, and especially bivalves, most of the data here in the map, you have those black dots, which are the data available. Most of the data is shells only. And eDNA detects only live specimens. So if you see all those black dots in the region of Paris, actually these are all data, shells only, and the presence was not of the species was not confirmed with eDNA. So this allows us to say that the species is probably extinct from the same drainage. Okay, this is what we published in 2023, but now we know that some populations are remaining, and even in the center of Paris, which was unexpected, because we have many more eDNA data now. But this is, I mean, an interesting result saying be careful of the distribution maps for freshwater mollusks. It's mostly shells, and shell may last for long. On the other hand, here in the Rhone drainage, we detected a species that was not known from this river, which is a large river, hard to sample, and the species was completely overlooked. This is what I illustrate here with two case studies. Dracena bugensis is an invasive species that was only known from the Meuse and the Moselle drainages in the north. And then I detected it in the Saône here, and we made a small paper saying, oh, it's getting into the Rhone River now. So we had this hatch distribution, and with eDNA, we now know that it's been completely overlooked in the Seine drainage, that it has completely colonized the whole Rhone, and it's even going to colonize the west side from Canos. Uniocrassus, which is a habitat-directed species and a natural 2000 species, was known only from the hatch distribution we have here. So everybody thought that there was no Crassus below the south of the Loire drainage and only in the upper Rhone. And we detected it here and in the whole course of the lower Rhone. So it means that here, Natura 2000 measures can be implemented for conservation of this endangered species, which had been completely overlooked for years in the lower Rhone drainage. Less optimistic, this is the distribution of two species. So here you have Pseudodendronta complanata that you have in England too, which was thought to live everywhere, but in the Mediterranean small rivers and the mountains. But from the literatures and the shell data we have, was supposed to be everywhere. eDNA showed that it's now restricted to northeastern France with only a few scattered populations elsewhere. And the situation is even worse for this one, Spharium solidum. As the name indicates, the shell is very solid, and you have a lot of shell data. But I've contacted all the people that share these records, the black dots, and none of them had seen it alive. And eDNA confirms that with only two populations here that subsist, and we now know that there's another one here in the lower Loire River. But this species is highly endangered, and no one has seen that. I mean, it's been completely overlooked for both of them. So I come back to this Pseudodendronta context. There used to be three subspecies in France: Red Cupeana, Ligerica, and Complanata complanata. So from historical data, it was all France except mountains and the Mediterranean drainage, as I said. Recent shell data, the ones that are on the museum site, for example, consider it's the northern half of France, and actual distribution from eDNA showed that it's been extirpated from about 75% of its historical range. And as I on the other slide, there's 3 isolated populations here, and the core population is restricted to the northeastern France. It's very Amsterdam, as I said, very, very rare. And this is confirmed from other European countries. I talked with European colleagues, and they said the species is not seen alive in most of its historical range. Now, this species resemble a lot juvenile Corbicula species. So I suspect that there's a competition between those two, which would explain a very rapid decline in the last 10, 20 years in all Europe. So in a nutshell, what we can remember for that is that freshwater bivalves are difficult to find and to identify. Most data consist of shell only, and misidentification are very common in the records. eDNA provides an efficient way to monitor freshwater bivalves with a higher detection probability and reliable identification. I can add also only live populations and not shells only. And large-scale DNA monitoring in France allowed us to update the species distribution and conservation status. And it's also a tool for early detection of invasive species. Okay, let's go to a completely different ecosystem. I will show examples from the French Guiana. So the Guiana Shield is a hotspot for freshwater biodiversity. It's well known. In a single river, the Marani River, you have up to 250 species of fish. And in this small territory, which is French Guiana, it's only this small place of the Guiana Shield. You have over 500 species of freshwater fish. It's the same for insects, very well known, but very few studies have been carried out in French Guiana for bivalves. And of course, it's hard to inventory. So there are very remote species places. So it's hard to get there. You have to use very small planes that will take you to small airports. So only the plane is challenging, especially for me who doesn't like plane at all. And then once you're there, you have hazard species like the electric eel or this torpedo. Both of them can be deathful. So it's very hard to survey rivers, especially as you can see the turbidity is very high. So it's very hard to survey freshwater mussels there, although there are some. In 2019, there was only 5 freshwater mussel species recorded in French Guiana, which was surprising given the big biodiversity of fish and insect that is documented there. So I had this big project, and we did eDNA samples everywhere, plus traditional methods. Here, the white dots are the places where we try to inventory and dive into the river to try to find some freshwater mussels. But the coverage with eDNA is very good. We have about 150 sampling sites there. So we managed to detect the species that were known. Of course, I had to do the reference database. So I had to collect all these species alive, sequence them, and make the reference database to be able to say that the eDNA sequences that we collected were this Castalia subcata, for example, Diplodon holzi, Diplodon uranosus, I'm missing one. And we had all these orphan sequences, some of them quite abundant. And all these, we didn't know what it was. So I made, well, you can see, you put them on the global phylogeny, you know that this is probably an Anodontites, and this is a Mycepetopodidae of some kind. So you know it's freshwater mussels, but the species are not identified. The frequencies are often, we don't know what it is. And for some of them, we have a lot of them, like in Kourou, 5,000, that's I mean, the species here is very abundant, and we don't know why it is. So we came back in 2023 and collected at least two of those, which are cryptic species. I mean, there's absolutely no morphological differences between Adondotites crispata and those two. This is why they've been completely overlooked by other malacologists. But the genetic distance is very high, and this one is even a new genus. We called it Pseudodontites because it's really like an Adondotites, but it's a new genus with over 8% divergence in CO1. And the other ones are still to be found. But, well, this is the kind of result we could have after this big sampling. We had only very, very scarce records of freshwater mussels, and now we have an atlas. Anodontites crispata. This is the new species that we describe, and we can even map the undescribed species based on environmental DNA data, like Mycetopodes SP1, SP2, SP3, which are still to be found. And as you can see, the distribution is very coherent. This one is probably endemic to the upper Oyapock. This one too. This one is only found in the Maroni, not in the Oyapock. This one can be found in both rivers. So we still have to go there and try to find live specimen in order to be able to describe there formally, because of course you need a longer sequence first to confirm this molecular data, and holotype, which we don't have. So only two of these cryptic species have been described so far. And this is also a very interesting results. Here you have, as you know, the main threats to these rivers is gold mining, and you can see the impact it can have on a small tributary here with all this orange flowing water, which is heavily concentrated in mercury, for example, and completely turbid. This has an impact on all the fauna. And here on those maps, the most impacted sites are in gray, and most of the impact is in the lower Maroni here, especially here. You can see it. And the upper Maroni is quite free. So as could be expected, the species richness is higher in all samples in the upper Maroni, which is in contradiction with the ecological model, because normally, on a river, you have a higher species richness downstream than upstream, but this is due to human impact. And what was new is the second mapping V, is that the intraspecific species richness is also higher in pristine areas. And this kind of result is only available with eDNA, because eDNA collects all the genetic diversity in the river, which you can't do with traditional methods unless you kill everything. So this was quite interesting because it's not documented, to my knowledge, that intraspecific richness can be affected by human impacts on the environment. Okay, so that the abstract of what I've said. We did eDNA. We found 6 species, and coming back to the site a couple of years later, we managed to find at least 2 of those cryptic species, and 4 are still to be found. That's it. Thank you, and I'm happy to answer any question.